Thank you. So successively, I'd like to present our recent experience on this first stimulation of the primary visual cortex in humans. This study is presented on behalf of the principal investigators, Yonghan Chung and Wonhye Lee and Seung-Sik Yoo. So functional MRI and PT have been used to characterize the spatial temporal features of the neural responses to first mediate the brain stimulation in anesthetized small animals. Yet, evidence of increased neural activity in the stimulated region, as well as the associated network-wide brain responses, remained unexamined in conscious humans. The study objectives are first, to examine the stimulatory effects of FOS on the primary visual cortex in humans. The visual perception, fMRI bold signal, cortical EEG responses were proved. And second, to examine the brain areas activated due to the stimulation to the V1 in conscious humans using simultaneous acquisition of fMRI. We also examined the safety profiles after the sonication to the V1. So in this study, 19 healthy volunteers participated, and in pre-screening procedure, fMRI CT scanning was performed to be used for FOS planning for targeting the V1. And the multiple FOS sessions were conducted in separate days for fMRI FOS session and EEG FOS session. The post-sonication safety examination was conducted, spanning 2-month period. This slide shows the schematic of the fMRI FOS setup and the MR-guided FOS targeting for the V1. Event-related fMRI was simultaneously conducted with FOS sonication, and 300 millisecond long stimulation was given 50 times every 13 seconds. These are the sonication parameters used based on our previous study, and single element FOS transducer, 270 kHz one was used. The sonication size of the FOS focus at full width half maximum was 3 mm in diameter and 17 mm in length. So upon sonicating the V1, 11 subjects reported the perception of a FOS beam. Most of the visual perception were described as a diffuse, amorphous, non-colored brightening of the entire visual fields without the presence of a retinotopical arrangement. A few subjects reported patterned FOS beam or colors in patterns. Other 8 subjects felt elicited FOS beam events only a few times or did not report any sensations. These are the individual and group level fMRI responses from the responsive subjects. The area showing activation in the V1 was spatially aligned with the sonication target. And we would like to note that FOS activated not only the sonicated V1, but also other brain regions in the primary visual pathway, visual association areas, and attention-related neural network. Photic stimulation elicited activations in LGN, V1, and fusiform, while the sham sonication did not elicit any activations. These are the EEG responses and acoustic simulation results. The FOS generated stimulation-specific EEG responses having P100 component that was also detected from the photic stimulation. And we used instant intensity of 16.6 watts per square centimeter ISPPA, and the simulated intensity profiles revealed that the in situ acoustic intensity at the target was about 3 watts per square centimeter, and the spatial deviation of focus from the target was about 3.3 millimeter in average. And post-FOS safety profile of 2-month follow-up revealed no abnormal findings across all subjects. So overall, FOS activated the sonicated brain area of the V1 as shown in fMRI data, and concurrently elicited the associated afferent sensory perception in the form of a FOS beam. Simultaneous fMRI activation revealed in conscious humans that FOS also activates the network of regions involving visual and higher order cognitive processes. Successful stimulation of the V1 was also supported by presence of evoked EEG potentials. And retrospective numerical simulation showed the individual variability in intensity as well as location of acoustic focus. Thank you for listening.
Thank you. Questions?
So I thought it was really interesting, the lack of spatial specificity in your phosphines. You said that basically they're diffuse and covering the entire field.
Yes.
Did you try basically to relate that to retinotopic mapping of the visual fields that...
What is the original topic of mapping?
Mapping of the visual fields that you did in the same subjects in the fMRI?
We used
I guess if you were to predict kind of what they would see, would it be like a full field, or were you expecting something more punctate?
Initial stage, we expect this kind of retinotropic kind of arrangement, but we didn't see the kind of results from this study. But we reference to the kind of fMRI data in the sonication planning at the initial stage. So it's kind of reference to the functional data.
Just interested in a couple of things. One, how long did the phosphines and the paresthesias last after the low energy sonication?
Very transient.
Okay.
Yes.
Did you try motor map or language mapping?
Language mapping?
Yeah.
No.
Okay. And then what kind of sonicator was that, and how was it guided exactly? And specifically, does that particular one have an FDA approval?
Before the starting of a post-sonication session, we mapped the functional visual area of each subject using kind of photic stimulation process. It's a kind of separate functional image mapping session we had. And based on the functional image data, we found the peak activation of the visual area from each subject. And we set as a target location. And based on that coordinate information, we just designed the fMRI process sessions.
Okay. Mm-hmm. Who makes the device, the sonicator device that you use?
In our lab.
It's homemade. Okay.
Yes.
Thank you.
I have a question to you. Do you know how much skull heating did you get during the sonication?
Skull heating. It's a good question. But we are using very low intensity. So we didn't expect kind of increase of temperature. But what we know is, based on the simulation result, the attenuation occurred at the skull interface was about 70%.
But you had 16 watt per square centimeter?
Oh, yes.
That's not too low.
Not too low. Okay.
So you don't have an idea how much they heat?
I don't have the specific idea about that.
Thank you.
Thank you.